Hi everyone, this is Meredith. We are going to get started in just a few minutes. Thanks so much for joining us today. So just hang tight and I'll be back in a little bit. Hi everyone, thanks again for coming. If you've just joined, I'm going to wait a couple more minutes before I get started. So yeah, thank you for spending your valuable time with us today, and I'll be back in a little bit. Good afternoon and good morning, good evening. I think I saw some of our users and colleagues from Asia on the line as well. So my name is Meredith Price, and I am the business manager for Imaris here at Oxford Instruments. Today, I'm going to tell you about our newest release, Imaris 9.7. I want to keep your attention, but at the same time, it is available online right now as well. Um, and for this release, we have focused on two separate stories, structure and dynamics. And the idea for both of these stories is that we're going to help you get more out of the images that you have. So we're going to give you even more information to help you understand the structure in your 3D images and 4D images, as well as understand the dynamics that are happening in your time-lapse images. So I'm going to take a little bit of time first to introduce Imaris in general to anyone that perhaps hasn't used Imaris before or doesn't know much about it, and then I'll dive into the structure and dynamics. Okay. So first off, Imaris is a family of software. We have Imaris proper, which is our flagship, our original software that performs visualization, analysis, and interpretation for 3D and 4D microscopy images. We also work on 2D images, but we're definitely best known for our 3D image visualization and analysis. Within that Imaris software, we have 10 application-based modules. And so those allow you to specifically work on things like tracing neurons or working to segment cells and the organelles within them. And then we've also spent a lot of time making sure that Imaris is big data capable and uses dynamic multi-resolution rendering to ensure that you're getting the fastest but highest quality rendering whenever you're working with those big data sets. Imaris has some siblings. It has Imaris Stitcher, which is a newer product where we're taking image tiles and aligning those and fusing them for you. So oftentimes, these can come from a variety of different microscopes, and they typically come together to create a very large data set. So again, our big data capable understanding and performance is important there. We also have Imaris File Converter. This is free. You can download it free from our website. Just go to the demo page, and you are able to convert any of the raw microscopy file formats, which we support over 50 of those, and convert those into Imaris files, which are automatically opened within Imaris. And so then we also have Imaris Viewer, which is another free software. It is a lot like Imaris. It now can become your raw microscopy image viewer. So you can use that to explore raw data sets as well as the Imaris files that you've gone in and analyzed. So again, that's free and also available from our website. So with this in mind, exactly what kinds of things does Imaris do? So I spoke about visualization, and um, as well as quantification or analysis. And so if we take a deeper look at visualization, these are the types of things where we're importing the files, we're converting those to IMS files, this Imaris file type, which is a multi-resolution pyramidal file type that allows us to work very easily through different resolutions, and to ensure that as you're looking at the highest resolution and you've zoomed in, Imaris is reacting and interactive just as fast as it can be. We also offer deconvolution. We have a number of different pre-processing steps, as well as stitching, and then you go on to actually rendering your data set. But don't forget that we have presentation tools. So we also allow you to export out snapshots of the work that you've done, as well as animations. We also perform quantification, and this is where this is another place along with rendering where Imaris really shines. And so we have a few different models, and you can see those up here. I need to give credit to Georgia, one of our salespeople based in France, who created these first slides. And so they're really nice because in this slide, what we're pointing out is that you have several different models that you can use. You can use them in different ways to analyze different types of objects that you see within your biological images. And so whether it's nuclei, which you can characterize using just spots to count them and find their positions, or if you want to use surfaces on cells or nuclei to give a better understanding of exactly what that volume is and that morphology. You can also do that kind of work using filament tracer, where you can trace out all the processes in this cell or trace a neuron and understand all of the different branches that you have, count those, and that sort of thing. So with integrating all of these different models, then you can go on to do things like tracking through time. You can classify different types of objects within your image to understand within all of those spots you found. Maybe some of them are expressing a transcription factor differently than others, and ensure that you're finding those. And then finally, we do have batch processing so that that work that you've done can be repeated in a reproducible way over and over again without you having to do all of those mouse clicks yourself. So that gives you an overview of what Imaris is offering already, but what are we introducing with 9.7? And so with 9.7, we are going deeper into spatial statistics. So for a long time, Imaris has presented you, as I said before when I was talking about spots, has presented you with the number of objects that it's detected in your image and the position of those objects. More recently, we've added in measurements for you to understand how far away those spots are from other objects in your image, whether it be a surface or something we call a reference frame. But what we haven't done is to really dig in a little bit deeper and help you understand if this if the positions of these spots and their localization with the image, if it's statistically significant or if it's significant relative to another object. And so the first question that this release will help you answer is, this is one that applies to the image over here on the right, where we have vessels in this reddish pink color, and then microglia nuclei have been detected, and now we see them labeled with these blue spots. And so can we say that the blue spots are found near the vessels? But more specifically, can we say that they're more frequently found close to the vessels than we would see in a random distribution of cells if we were to run a simulation, let's say? And so that's one of the big questions that Imaris is going after. If we have a reference, and in this case it's the vessels, but it could also be a tumor, it could be different types of cells, are we seeing some sort of attraction where the blue cells in this case are found more often closer to those vessels than you would expect with a random distribution? So that's a big question to ask. How is Imaris actually going in and answering that question? And so I'm going to go through some slides and then I'll show you what the results actually look like in Imaris. I'm going to get through the spatial, all of the spatial stuff first, and then go over into Imaris. So how is Imaris answering this big question? And really the first thing you have to do isn't that big of a deal. It's something that you're doing all the time in Imaris. You're going to find your reference and have Imaris identify that reference as a surface. So in the last case with the vessels, you identify the vessels as a surface. And you need to make sure that as you go through that creation wizard, you have a checkbox for object-object interactions checked on. That's going to be on by default for the first few objects that you, or components that you find. Once you've found that surface, then go in and find your objects, or your, in my example, the blue cells, and you find those as spots. And then Imaris can assess if those, if the objects found inside or outside or both, how far away they are from that reference. So what's the shortest distance from the spots to the surfaces? Now, an example like I had before, it's hard to know from this image, is this are the blue cells distributed randomly, or is there some sort of attraction perhaps to those vessels? And so to help you understand that, instead of just dumping a lot of spreadsheets onto you with lots of values, instead, we're going to go through and create some plots for you. So we're going to automatically create a distribution plot, and we're going to plot the number of observed objects as a function of their distance from the surface, as well as running random simulations in the background automatically. Imaris does this for you without you having to do anything, and also plotting that complete spatial randomness, plotting that as well, the plot of these 1,000 random simulations. So what does that look like? So again, first you find your reference, your surfaces, then you find your spots, your objects, and then you go into Imaris Vantage, which is our plotting view, and you will automatically, within the spatial view, it's a new icon you'll find, you'll automatically get this plot. So what is it telling us? So I'm just looking outside and this is an option that you'll see within Vantage. I'm just looking from the edge of these vessels out. And so you can imagine that the edge of the vessel is here at the 0 mark. And now Imaris is plotting how many spots are within a given range, and the resolution you can see is about 1.7 microns. So how many spots are within 0 to 1.7, and you get this value here. And so it goes on and does this as it continues to go out in steps of about 1.7 microns, and is counting how many spots you have. And so this is a density plot, so we're looking at percentages. But it's based on the original counts. And so you can see that there are a lot of spots within, let's say, 0 to 20 microns around these vessels. Okay, so that's the observed distribution that we see. But what about these simulations that I spoke about? So you can also turn on the option to show what the simulations look like. And so you see that in the dashed line, you see the median value over the course of these 1,000 simulations. You see that median value as well as a confidence interval for those simulations. And so we can say for sure that the what we saw in the observed or your original image, there's definitely more of an attraction or more spots found from 0 to like 18 microns than we see in the simulation, in the random distribution. Okay. You can also see that because of the shift to attraction in the lower distance values, we have a shift to repulsion, where we're underneath the curve. We have less spots in this range from about 18 microns over to, let's say, 40 or a little bit more. Now, picking those numbers to say where your line is, where you see the attraction or where you see it move from attraction to repulsion, that can be something that you want a little bit of help with. And so Imaris also goes in and adds these attraction distance bars that help you understand where that switch is happening and gives you a way of reporting exactly what those values are and describing that to the people that you're presenting this work to. So this type of work is definitely happening within the biological world, but it's one of those things that's more difficult to do because no one software can take you from a raw image all the way to these plots. And at least not in 3D and over time. And so now we're offering that here, and it's all completely automatic for you. So that's one question. The next question is, okay, but what if my label is diffuse and I can't use spots? And we see this sometimes. This one actually is pretty punctate, but it's messy. And so you can see nuclei here in pink with this cytoplasmic, something in the cytoplasm in green. And so are these green dots, and is the green intensity really just that green label? Is it found closer to the nuclei, or is it evenly distributed throughout the cell? And so this often comes up, and that's why I use this example with nuclei, because people want to look at if a protein has a perinuclear localization, or if disruption of the nuclear import is causing sort of a pileup right around the nucleus and that sort of thing. So there's nice experiments where this can help you as well. And so how do we approach this question and answering this question in Imaris? Again, first you find your reference. In this case, for my image, it would be the nuclei, and find those as surfaces. Make sure you've got those object-object interactions checkbox on. And then you just go straight into Vantage, and there will just automatically be a distribution plot created for you where it's looking at the average intensity as a function of the distance from the surface. Okay, so what does that look like with pictures and not just words? So you can see here that I've found the nuclei. They're still pink, but they're surfaces now. So they're the solid object, our model. And when you go into Vantage and go into Spatial View and pick the intensity plot, what you'll see is that now you're seeing lines illustrating the average intensity as you go out from the nucleus. And so again, Imaris is telling you what the resolution is on the x-axis, and so it's about 1 micron again. This is a function of the resolution of your image. And what we can see is that the nucleus channel is in blue, and of course it has a steep. It has it's the line is very steep right here because that's at the edge of the nucleus where I created the surface, so that makes sense. But we can also see that the vesicles channel, which is labeled in red here but matches the green channel over here, we can see that it starts off high and then goes down as a function of distance away from the surface. And so we do see that there's an accumulation in this image of the green channel closer to the nucleus. And so again, these plots are made automatically for you and give you a quick look at the answer that you're looking for. These also work over time and can be a great way of showing changes through time when it comes to accumulation of a protein or changes in the distribution of cells or that sort of thing. Okay. So we have one last type of analysis that I think is really important within Imaris to help you better understand the distribution, the spatial distribution of the labels and the objects that you find in your image. And so that question is, do my proteins overlap? And if they do, how much do they overlap, or to what extent are they overlapping? And what you're seeing in the background of this image right here is are two separate proteins. One is just in this white or gray channel. It's transparent or semi-transparent. And then you see another protein, and that's all the colorful ones, that has been identified and color-coded by how much it overlaps the stuff in gray, the protein in gray. And so if it overlaps none at all, it's this dark blue. If it overlaps more so, you're going to start to see the reds and oranges and yellows, and my color scale bar is missing from this image. But and so not only do we create this color coding for you, but we'll also give you a report on that. So how do you go about doing that in Imaris? Well, by its nature, you need to have two separate surfaces, one for protein A and one for protein B. You can have more. You can go crazy if you want. If you have lots of labels and maybe doing multiplexing. But let's start with two. And then after you find both of those surfaces and make sure that that object-object interactions is checked on, then you can simply go into the statistics tab, where under detailed and under specific values, you'll find two new measurements, or what we call statistics, and overlapped volume ratio is the first one, and overlapped volume is the second one, sorry. And so then for each and every surface, it will tell you how much it's overlapping with the surfaces of the other type. Okay, so those are, how am I doing on time? Okay, those are the three big new things that we've added to help you understand the spatial landscape, the structure of the objects within your image. And I just want to take a little bit of time and show you what this looks like in Imaris. Okay. So right now, Imaris was already open. I'm within the arena view. I switched from observing my really messy folders where I keep all the data to now just looking at a very specific folder just for today's launch. So you don't see how how messy I am usually. Okay, so the aspect I really want to show you are these plots. So from that first question, are the blue cells found more often, or are they found closer to the vessels more often than you would expect from a random distribution? So again, I've gone in here and I've created vessels as surfaces, and so they're in red. I've got the blue cells as spots. And I'm not going to go through the steps for doing that because I want to spend more time on showing you what the Vantage plot looks like. So when you go into Vantage, make sure that you pick the Spatial View button. This is a new button, a new view that you have within Imaris now. It does require the Vantage module. When you go in, you need to be on this option where it has little spots, little orange spots there, and that means that you're going to look at this type of analysis in the plot. Right now, I am looking at a probability density plot. I'm going to first take away some of the extras here. So what I'm looking at is the observed line of how many spots are within each of these sort of rings or shells going out from the surface. And all of that data is down here. You can look at that in the table. It's also available within the spots tab or within the spots object on the statistics tab in the Surpass view. Okay, now you may ask, okay, Meredith, there are 4 plots. So we wanted to make sure that we took care of and accommodated all of the different types of plots that people might want to look at, as well as the numbers that go along with those. So the raw numbers are going to be in this histogram count plot, and you can see that this can be quite noisy. And so for that reason, we've offered you the probability density plot, which does perform some smoothing. And it gives you a better idea of what's happening. We also know that oftentimes people want to not look at the number of objects in each individual ring or shell around the surface, but to add them up and create a cumulative plot. And so this is what that looks like. From 0 to 10, you're going to have all of these objects, or you're going to have from 0 to 10, you can see that there are 85 different spots found within 0 to 10. And so you can put your mouse along the plot and see the numbers as they change. And then, of course, we've got a smoothed version of this cumulative plot, and this is the CDF plot as well, so you have that. Now, this information on its own, yes, it tells you about what you observe, but it doesn't tell you much about whether that is different from random or not. And so now what you can do is add in this complete spatial random generated data. So again, this is 1,000 simulations using the exact number of spots that are in your image, and that is then plotted. And so the median value over the course of those 1,000 simulations is plotted with the dashed line, and then we have a confidence interval in that shaded region. So now we can see that in this region, because our observed line, the non-dashed line, is above the dashed line, then we can say there's more spots closer to the surface or to the edge of that surface. And so we see an attraction there. We don't know why there's an attraction, but we do see an attraction. I think it oftentimes comes down to preference, which one you like. I do prefer the probability density plot. One reason that I prefer using that is because you can actually see at which point the observed line starts to cross over the randomly generated line. And so I do like to see that, and then you'll often see that the attraction distance is found, the attraction distance that we label for you to help you understand what's happening in your plots is often very close to where that crossover is. But it is based on the raw data that you see in that histogram plot before any smoothing happens. So there's a couple of other options I want to show you. You can look both inside and outside of the surface at the same time. That's a different round of simulations that are run. And you can also look just inside the surface. And so there's actually 3 different rounds of simulations run depending on, and 3 different rounds run, and which one is shown depends on which object or which option you're choosing over here. There are some options over here under the frame tab. So if you've got Imaris 9.7, or if you test it out now that you see these features, go over here and play around a little bit. You might hate the minor grid. Go ahead and take that away if you want to. Okay. Okay. So hopefully that gives everyone a good introduction to these plots. The intensity plots are pretty similar, and then the overlap information is, or the overlap statistic is straightforward under that statistics tab. I do want to go back and really quickly just make sure everyone knows about the new tab within the statistics tab as well. And so you will see that there's new statistics that we use to create the plot. Those are available within the Surpass view and are this new additional tab called Spatial within statistics. Okay. So yes, you can look at the raw data if you want to. That's the point there. Okay, so now we're going to shift gears and start to talk about time-lapse images. So I'm going to go back to PowerPoint. So I said at the beginning that the focus of this release was really to dig in further into both structure as well as dynamics. And for dynamics, we're introducing a new way of looking at events that happen within your time lapse, and it's called event time. And again, this results in new plots within the Vantage view and helps you get a better handle on what's happening in those time lapses. So here I have a C. elegans early embryo, which is going through cell division. So we see the histones labeled with GFP. And within this image, there's different things happening. And even if it's just this one channel, there are things that you can study and measure, of course. And one thing that you might be interested in is how are those chromosomes that are being divided, how are those moving during anaphase? Are those ejection forces really pushing and pulling them apart quickly, or are they kind of lazy and not moving very fast? So that's a question that you can answer here, but with this image, but really with our new feature, being able to look at event time, you can look at all sorts of different changes that are occurring relative to a cell division. And so again, how does Imaris do that? How is it, what's the process for being able to take a look at these questions more deeply? So first you'll have your raw image or your raw time lapse. You'll go in and both segment as well as track your objects of interest. In this case, it's the chromosomes. And so we can see that there are cell divisions. Those get automatically detected using our lineage algorithm for tracking. They are automatically labeled as an event when you use the lineage tracking, and they can automatically be labeled with a color for you. So in this case, that's red. And then once you've done that, if you go into Vantage, you will automatically have a plot created for you. And it's going to use a new measurement called event time, where for each object that's labeled as an event, so in this case, it's the ball of chromosomes right before they divide. So it's the time point right before we see that they've split. They've been labeled as a track split. And for event time, right at that time point, we label it as 0 event time. And what that means is that even though there's divisions happening at all different times in the raw image, once we've segmented and tracked it, we can synchronize our plot such that we're at this 0 time point, we're looking at just the cell divisions. So Imaris is able to classify by cell division, calling it this track split. It's then taking a portion of the track and displaying it for you, but it's doing that for all the different times that you see a track split and showing those here. And so what you're seeing is with event time, this is before cell division over here on the left, to the left of the 0. It's after the cell division on the right-hand side. This is in seconds. And then you have speed for the y-axis. And so we're looking at instantaneous speed. How fast are those chromosomes that I've labeled with spots moving just after that cell division? And for a nice, healthy, early C. elegans embryo, you want to see them move very quickly away from each other. And so that's what we see. We see this increase in speed right after the cell division, and then it starts to slow down after that. And so we do see this really obvious change. It's a nice positive control to make sure we're seeing the right stuff. Okay, so that is one thing or one way event time can be used. And some of you might be like, I don't work on cell division. What about me? I do time lapse, but I don't work on cell division. Okay, although I love cell division. So there's, we can extend this idea of events to other changes that we see within our images. If we can detect it, then you'll be able to create these event plots around it. So here is an image where we have parts of a vessel labeled in red. And so this is the junctions for the cells that are creating the vessel. And then we have other cells that are migrating around it. Sorry, I don't have a video. And the ones in this whitish gray are pretty far away. It's hard to tell because, of course, it's a 3D image, but the white ones are farther away, whereas the blue ones are very close by. So do we see a change in, let's say, the speed or the acceleration of the cells as they touch these junctional proteins of the vessel? And so again, that's another thing that you can test using this event time and these time plots. So so that's an example of contact. It could also be that you're looking at a protein turning on, and do you see an effect in a different protein? So the red channel increases, you classify that as an event, and then for the next step, you want to see if you see a decrease in another channel, or if you see more puncta in another channel, something like that. So how do we go about answering this question within Imaris? Here's the our example image, and you're going to go in and make sure you've segmented both the vessels as well as the cells that are migrating. And you need to classify them. So you need to make sure that you're classifying your cells that are migrating. You need to classify them based on the event you care about. So the event in my case is that they've gotten close to or touching the vessel. And then once you've done that, you can move over and automatically see this plot available to you within the Vantage view. And so in this case, what we're seeing is again, we've synchronized everything, all of the portions of the track where you see the event that contact has happened. We've taken all of those portions of the track and we're looking at them as a group. And so we see the median value in the white line, and we see the confidence interval or sorry, we see the quartiles um, for in gray. And so we can see that, yes, around the event time, there is an increase in acceleration, but I'm not, at this point, I can't say that that is something that's significant and different. So more work needs to be done, but it, it does suggest that there may be something there. So further work to be done after looking at one image. But this is something that you can do, of course, with multiple images and look at. And so that's another way of looking at things. And so we call these events class change events because you would set up different different classes within Imaris, and that is during the creation wizard, and I'll show you that in a little bit so that you understand. You set up different classes and then you assign events to when they change from one class to another. Okay, now you can say, but I don't have any of that happening, Meredith, but I am applying a drug or I'm bleaching something within my image. So do you have events for me? And yes, we do have events for you. And I realized my number didn't change. That's okay. So this is number 3 for events. And these events we call image events. So they're oftentimes extrinsic things that are happening, like a laser ablating something or bleaching or delivering a drug. And so this is a case of, sorry, this image is a case where we do have bleaching, and what you're seeing is that, yep, in this little region, there were, it, in the beginning, it was, it had lots of signal, like the rest of this tissue in here. A bleaching happened right here. I created an ROI for this region and tracked that through time. And then I said, oh, I know that the bleaching happened at frame 6. And so I can use this new star icon to add an image event. So this means that an event is happening to everything in the image. It's not specific to a track. And then I can go in and look at dynamics against that event time. And in this case, it's the dynamics of the intensity change. So we can see the intensity was nice and high before the event. As the event happened, it dropped down quite low, although not completely to 0, but quite low. You can see that. And then it started to gain again. And we saw the recovery of that fluorescence within that purplish pink region. And so this gives you a nice way of looking at those types of events, which has been something that traditionally Imaris wasn't used for. And so this does expand what Imaris can be used for, especially if you're in something like a core facility and wanting to have as many people using the same software as possible. This is a new thing for us. Okay. So I've got a little bit more time. So let me go in and show you what some of this would look like. So I'm going to take the C. elegans example. So here's my embryo. Let's see if you'll play. You might not. Oop. I have the beta running, so that's what happened right there. Sorry for the momentary hiccup. Okay, what I'm going to do in the meantime, since I can hear all of my hard drives booting up in the background and Imaris wants to connect to all of them first, is I'm going to go through and just tell you the other features, and then I'll go back to the event stuff to show you inside of Imaris. So we do have some more things. We have a contact duration statistic. So this has been asked about previously. We implemented it in this release. And so you will see, or sorry, I put contact duration, I mean class duration. It's oftentimes used for assessing contact, but it's class duration. So if you want to see how long a cell is bright green for before it dims out, you can set thresholds to look just at the bright green and find those values. We also made the distance transform native, so you can find that within the mask window for surfaces and spots. We also have some other options within the time plots, which I'll show you in just a second when I back out and go into Imaris, where you can look at time since track start, and that will allow you to create median squared displacement plots, which it will be a really nice feature for a lot of the people working on motility and using Imaris. And then you will find, and some of you may have already if you've downloaded Imaris, you will find that there is new licensing. So it should be happening in the background for you, but if you have any hiccups with that, just like I had a hiccup with Imaris, feel free to reach out to our support teams. Okay, so let me go back to Imaris. Things have calmed down. Okay. Okay, so I've gone through and segmented and tracked this data set. Again, I've used the lineage algorithm, so it has created events at every single track split where a cell divided. And if I go to Vantage and then go to Time View, so you see a little stopwatch, what you'll see is that if I plot by event time on the x-axis, and you do have other measurements that you can use here, so you can just do straight time, time since track start, like I just mentioned, as well as some other event time type of measurements. So time since an event or time to an event, and there's more information about those in our release notes as well as within our reference manual. But you can choose any of those, anything that's time-based, and then for the y-axis, you have all of those different measurements, object-based measurements that are available to you. So you could look at spatial information like average distance to nearest neighbors, or displacement measurements, that sort of thing. But in this case, of course, I'm looking at speed, where we see that nice change in how the chromosomes are separating from one another. Again, we are showing the quartiles, so I can turn those on and off. That helps to give you an idea of what the variation you see within your image, of course. And then you're getting box plots, which are showing you for all the tracks that are included in this plot, what the quartiles look like for those, and their min and max for the range. Over here, what I haven't shown you yet or mentioned yet is that there is a histogram here, and what it's doing is it's giving you bounds on what you're seeing within the plot. So this is all event time, and I've set it up to look from negative 300 something seconds to positive 332 seconds. On either side of the 0 or the event time. I can broaden that if I want. You can see that out here, there's not too many tracks that are included, so I won't include those, and then maybe go out to here. So left click on the left side, right click on the right side, and I can expand that out and see that, yeah, there are some other increases in speed over here earlier in the tracks. And so you might want to go in and investigate what that is and what that means. And you can do that because down here in the detailed tab, you can sort based on that event time and see what those values are for each of the objects that have been tracked and are shown in this plot. Okay. Like I said before, with spatial plots, you have a bunch of different options. I would recommend that you go in and play with these. You can change line widths and that sort of thing. You can also, when you see these squares, if you hover over it, we have a nice tooltip telling you that you can edit edit the color. Most of the time within the time and spatial views, you can edit these colors and you get these nice color bars where you can go in and change the color if you wish. Okay, now let's see. At this point, we're running low on time, so I want to make sure I show you one more thing, and that's how to set up these class change events. So I'm going to open up this data set. And I've already gone in and segmented surfaces. And I've also gone in and segmented and classified and created events for the spots. And what I want to do now is actually go back into the creation wizard, but just jump straight to the classification step to help you understand how to set up these classes and then what that means for events and the next steps to do that. So I've clicked on the classification button there so I can skip all of the segmentation and tracking. And now I'm in the classification step for the spots. And what I'm looking at is shortest distance to surface. And so for all of the gray spots that you see here, Imaris is calculating how far away they are from the edge of the surface. And then I can set up classes within the creation wizard based on that. And so you can move the slider, move where I am so you can see the number, move the slider. I'm putting it at about the radius of those spots. And so then once you have your classes set up, which you can do multiple sets and multiple classes if you wish, you don't have to do just one, you can add, or just two, you can add in more classes. Then you can move on to events. And the way that it is, it will start off being presented to you like this: class change, any to any. So from any class to any class, it will create an event whenever any class changes occur. So I want to be more specific than that. And I think most of the time you will want to be more specific than this too. This is just a really nice default setup in the beginning. So when you click on any, it will say from what class. So I want to say from far away. So that was class B, and you can change these names if you need that to help you. To when the cells get close, I want that to be my event. When the cells go from being far away and then they move closer, that's an event. And so that's how you set up that event. You can go in and maybe say, I want to look at the opposite. I want to say when they go from being close to far away, that's also an event. You can totally do that. It is incredibly flexible, and it just depends on what your experiment is and what you're interested in looking at. So then once you've done that, you just finish the creation wizard and then move over to Vantage. Let it do some calculations. It needs to do some more math because I moved things. And then you will have those event plots set up for you, and it doesn't take too long for that to do. And so much of this is completely automated so that you don't have to go in and do this act of synchronizing tracks based on their event times. And so now you can see that I have, I don't want to look at surfaces, I just want to look at spots. Now you can see that I have different event times available to me. I have event time from class A to class B and from class B to class A. I can put bounds on the times that I'm looking at around the event time and look to see if acceleration changes within or around the event time. So it is very simple. If you're interested in doing this kind of work and you're a current Imaris customer, definitely reach out to us and we can help you with that, as well as with the spatial stuff. So I'm going to go back into PowerPoint, and I just want to thank a few people. So we got our images from a collection of really helpful customers. Thank you so much to everyone that's listed here. We have some great images that we love using. And then on the spatial statistics side, we've gotten a lot of help and we're inspired by the work of Alvaro and Caesar. So thank you so much for helping us with that and giving great feedback on the work that we had done earlier on. And within the release notes, you can look up their paper and read their paper, which inspired these spatial stats plots. I also want to thank the marketing team as well as the sales and support teams and the development team, and of course Anna, my right-hand woman, for helping with all of that goes into creating an Imaris version and releasing it out into the world. So yes, anyway, I am going to now stop sharing my screen so I can look at questions and answer some of the questions that you guys may have had as I went through. So let's see.
Thank you, Meredith, for fantastic webinar. So maybe to make it easier, I will read some of the questions to you, especially the ones which were repeated a few times.
Yeah, that sounds good.
Right. So important one, if we go back to spatial view in Vantage, the question you were always using spots model to detect objects. Can you also measure the distance between surface and surface using this tool? And if
Okay.
not, why not? Because I think it causes some, maybe a little bit of misunderstanding. This method is pretty specific, so could you maybe please tell a little more about that?
Okay, so good question. Thanks, Anna. So yes, it is for creating the spatial stats plot and for using the simulations and looking at if there's a difference to random, you do need to use one surface and one spots object. It will look at those together and only those. It has to do with speed and it largely has to do with speed. We're running a bunch of simulations in the background and calculating the shortest distance from the spots to the surfaces for all of those simulations. And so in those cases, we want to make this fast and we don't want to make it burdensome for you. So we have limited it to using spots and how far away they are from a reference surface object or component. But I understand that it might be limiting. One thing that you can do is that, of course, you can go in and for these microglia that we found using spots, so you can go in and reconstruct those as surfaces, and you can get the raw information of the shortest distance from the vessels to those microglia, get that shortest distance measurement for each of the microglia. However, it will not be used to create the spatial stats plot. Only spots objects will be used there. So you can go in and do that with two surfaces, but the extra steps of going into this spatial stats plot won't be there. Now, this is one of those cases where I do expect a lot of feedback from customers about the spatial stats plot and what we've included and what we haven't included. So definitely tell us what would be useful for you. Let your supporters and salespeople know. And yeah, we'll see where we go from here. Okay.
All right, thank you. I think it also might be important to mention that we actually compare with random distribution. And here in this model, I think the random distribution of spots is, let's say, easier or more simple to create as a comparison. But
yes, right. It's a speed. Yes. Yeah.
All right, we have a lot of questions. So maybe okay. So there was one repeat because I was answering the questions during the live session, but there were more coming in towards the end. So one question, maybe it's something do we do or do we not do? So we were mentioning the volume overlap. Is there also a way to measure the contact area of the surfaces?
No. There is someone has made an extension, which is a plugin, if you have the XT module to look at contact surface area, but we don't have that natively within Imaris. It is something that I'm personally interested in. I've helped a number of users when I was a salesperson to do that sort of work. It's something that I was interested in my former scientific life. Um, but we don't have that within Imaris yet, but it is definitely an interesting next step that we could take.
Right. So you also had some questions about the export of the plots. Is this
Yes,
only the,
thank
yeah.
you. So I forgot something. So thank you for these wonderful questions about this. So we do have the ability to take snapshots of these plots. And so you can see that. I'll take a snapshot and we'll get that out. You can, just like within the main or Imaris view, the Surpass view, you can change the size and the resolution and the type of export. But we also knew for a long time that for plots like this, as well as within um, yeah, oops, sorry, that for plots like this, people want the vectors to be able to take this into something like Adobe Illustrator to be able to get the plot from Imaris, but maybe change the font or change the colors and that sort of thing. So you can export an SVG file and it just prompts you to save that out. But so you can play along with that as well. Is that?
Yeah, I think that was it. Another maybe the option to we do have an option to export all the numbers as well if anybody wants to plot it outside.
Yes.
So
So again, you can find that within
yeah,
Vantage, but also under the Stats tab and the Spatial tab. You have
right.
all these different things. Yep.
We had one recurring question as well. Can you load two datasets and compare them in the spatial view plot? Let's say
Ah,
two different options like control and a sample.
okay. So there are some different ways of doing this, and I think maybe that is something that we should probably make a tutorial about. So I will save that and we'll get you guys a tutorial. There's a few different ways to do it, and I think I need to sit down and make sure the best way of doing it for you. But batch tools, some of those can work as well as just using our add image function here to be able to work with two images at the same time. Yeah.
Okay. Right. There was also a question about providing some examples of contact duration, but I'm not sure if we still have time for some demo or we cover it later.
Yes. So I can just quickly, maybe not, yes, it could be in this case, contact duration because I set it up to be that they're close or contacting. If they're in, these spots are in red if they're close or contacting the vessel labeling, so that you see in red as well. And so what you'll see is if you go to detailed for the spots, if you go to detailed and then specific values, you will see class duration and you need to pick the correct class. And so this is class A, and you'll see what if they are in contact, what the duration of that current contact is altogether. So this one, where are you? Let me move my GoToWebinar bar. You are hiding from me. This one, that spot, yeah. It is in contact with the vessel labeling for 1,560 seconds. And you can go in and you can go into the stats export and find out when that starts and when that ends. Yeah. Any other questions?
I think we can. I think for the recurring question, this was most of this. I think the other question was about the histogram and the resolution of the x-axis. So
Yes.
the space between them. So can you adjust it?
It it is available within advanced preferences to adjust it. I probably should not have even said that because someone will probably go and look, and I would want you to speak with our support team before you adjust anything within advanced preferences, but there are some places where you can change things within there. Again, it's initially the default is based off of the size of your voxels, and from there yeah, and from there it is possible to change, but we don't often recommend it. But yeah.
Okay. Maybe one more additional question about spatial view, because I
Okay.
think most of our, I'm not sure if you showed it, most of our examples were related to the intensity or distribution outside the surface. Is there also an option to compare with random distribution inside an object?
Yes. So in this case, in this image that I'm showing right now, I'm showing you the nuclei and the cytoplasmic label, but I could also show you the cells that with this semi-transparent surface, and we could look at where these vesicles are in the using the spots object uh, within those cells. And so I can then go to Vantage and go to Spatial View and turn off nuclei. Oop, let me go to turn off nuclei, turn on cells, and now I'm comparing where the vesicles are with the simulations. So again, the simulations have the confidence interval around them, whereas the observed won't. And what we're looking at is this edge of the surface is over here on the right-hand side, and negative values yeah, go lower as we go to the left. And so we're seeing that there's a decrease of these vesicles relative to what you would expect from the simulations. There's a decrease closer to the edge of the surface. And as you get more and more towards the center, you do see an increase of those, which correlates with the fact that we see them attracted to the nuclei. In this case as well. So yes, you can go inside as well as outside, and those options are over here on the left-hand side within the spatial stats view.
Okay, thanks. And an important one, what about batch mode? Are all these analyses available in batch?
Okay, so all of the computations are available within batch. Yes.
Okay, and maybe to wrap up, an important one, as you're always or often using the sentence that Imaris automatically calculates some numbers, which might raise a question. So, what about publications? What about papers? Are all these measurements documented? Can users learn how the analysis is done to make sure they can publish it in methods?
Yes, so I recommend that everyone, when you go online to our homepage, imaris.com, you'll see a big image to take you to the What's New page. Or from the homepage, you can go and look at, you can search for our release notes. And so I would recommend, if you're using these new features, to go through and read those release notes. And in addition, we have information about all of these features found within our reference manual, which you can find under Help. And you click on that, and it's an HTML file that you can then search through and browse through. The reference manual, as well as the quick start tutorial guide, are also PDFs that are included with your installation whenever you install a new version of Imaris.
Okay, thank you. Thank you so much. It was really great webinar. There were a lot of thank you comments as well, and we will
Cool.
try to answer all the, if any of your questions were unanswered, we will answer them later.
Awesome. Thank you, Anna. Thank you, everyone, for joining us today. Have fun with this. Let us know what you think, and let us know if you have any questions. We'll have lots of new tutorials up on the What's New page and within our Learning Center on our homepage, again, imaris.com, within later today and within the next couple of days. So thanks for joining us. I know you're busy, and enjoy the rest of your day. Take care.